Gene and cell therapies are becoming key treatments for many major diseases. As one of the most important gene transfer vectors, lentiviral vectors are widely used in GCT. However, the production of lentiviral vectors is often low in yield, with low consistency, and difficult to scale up the production process, making related drugs unstable and costly. To overcome these problems, Eureka Bio has developed an innovative lentivirus production technology, the EuLV system, which uses stable producer cell lines with serum-free suspension culture for scalable manufacture of lentiviral vectors. The traditional manufacture process of the lentiviral vector uses transient transfecting packaging cells with plasmids containing the packaging gene of the virus. In this process, the cells need to be cultured and expanded adherently. After reaching a certain cell number, the transfection reagent and the plasmids containing the packaging genes and target gene are added to the cells. In these cells, the viral genes carried by these plasmids transiently express the relevant proteins for assembling lentiviral vectors, and after assembly, viral vectors are secreted into the culture medium. Eureka Bio's innovative EuLV lentivirus production system stably inserts all the lentiviral packaging genes and the target gene into the genome of the 293 T cell to form a stable production cell line. The expression of each virus packaging gene and the viral vector is regulated through the inducible expression system. With serum-free and suspension culture, the production cell line can be cultured in a large-scale, high-density bioreactor or fermenter. When the cell number reaches the requirements, the inducer is added so that the stable producer cell line can massively produce lentiviral vectors. Using the EuLV system, plasmid transient transfection is no longer required during the whole lentiviral vector production process. When using plasmid transient transfection to produce lentiviral vectors, the number and proportion of plasmids in each packaging cell cannot be controlled and are different during the production process. Therefore, the consistency of the lentiviral vector produced by this method cannot be guaranteed, resulting a low lentiviral vector-specific activity, usually from 1 to 250, to 1 to 100. The lentiviral vector produced by the EuLV system has the advantage of high consistency and high specific activity because all producer cells are derived from a single monoclonal cell. In the large-scale production process, transient transfection lentivirus production requires multiple GMP production lines. Firstly, several plasmid production lines are needed. Next, the 3 or 4 plasmids required are produced through bacterial fermentation, plasmid purification, and other steps. Then, a lentiviral vector production line is needed. Finally, through recovery, amplification, then transient transfection, the plasmids produced in the first step together with the transfection reagents to complete the lentiviral vector production. When using the innovative EuLV system, plasmids and transfection reagents are no longer needed. Besides, the related GMP production facilities are no longer needed. The EuLV system can directly produce the lentiviral vector by adding inducer to the amplified high-density producer cell culture to produce lentiviral vectors. Compared with the transient transfection method, the EuLV system reduces more than 50% of the production steps and processes, and the unit volume expression of the lentiviral vector can be 100 times higher and ultimately saves more than 80% of the production cost. And it has a great advantage for a further scale-up. If you are interested in the EuLV system, please feel free to contact us.